Good morning, everyone. Once again, this is Okib Ajiteji from Ibadan Bacteriophage research team. Welcome to today's session. I will be looking at a continuation of our 2022 World Phage Week webinar series. So the theme for this year, for those that were not here yesterday, was emerging prospects in bacteriophage use and in clinical phage therapy. So today we'll be having Professor Jon Iredell. He is a director and chief investigator from Phage Australia. He's also a professor of medicine and microbiology at the University of Sydney. So we'll be taking us on phage biobank concepts, a national network to facilitate clinical phage therapy. So before we move on, let me just swiftly read a brief citation on Professor Jon Iredell. Professor Jon Iredell is a physician and microbiologist based at Westmead Hospital and the University of Sydney. His research group works on infections in the critically ill and in antimicrobial resistance, and has been continuously funded by Australia's National Health and Medical Research Council for more than 15 years. Recent work in the laboratory has focused on new solutions for antimicrobial resistant infections, including plasmid displacement approaches and bacteriophage therapy. Former affiliations are one, co-joint professor of medicine and microbiology, Sydney Medical School and Sydney ID. Is a director of Center for Infectious Diseases and Microbiology at the Westmead Institute for Medical Research. He's also a senior pathologist at New South Wales Pathology and Institute of Clinical Pathology and Medical Research. Can everyone join me in welcoming Professor Jon Iredell?
Thanks very much, Akib. It's a real pleasure to be here, and thank you very much for the invitation. I had trouble sharing slides, so Akib, are you going to go through those slides for me, or can I control them from here?
I'll go through them for you, sir.
Oh, I'm sorry to do that to you. Well, that's good of you. So I apologize, everyone, for my disorganization. But again, thank you for the invitation. So I might start with the first slide, please. I don't need to tell this audience much about antibiotic resistance, I don't think. We accept that antibiotics have really transformed healthcare, and there is a lot of concern that antibiotics are becoming less and less useful to us as antimicrobial resistance increases. The Jim O'Neill's review for the UK government points out that they are soon going to be the #1 cause of death, more than heart disease and more than cancer. And certainly our colleagues in India and our close colleagues in South Asia are facing this problem already. So we know, for example, that in India, if you have leukemia, you're more likely to die of antibiotic-resistant infection than of cancer. Next slide, please. And I look at your own national action plan and compare it with ours, and I see a lot of similarities. We, like you, focus on surveillance and understanding antimicrobial resistance, increasing awareness amongst health consumers and prescribers. You were in front of us in terms of recognizing the importance of One Health. So we have been a little bit focused on human health, but have only recently moved to a more comprehensive approach to surveillance and research. I've underlined your next 3 objectives because they're similar to ours and because they're important in terms of the capacity for bacteriophage to address them. Next slide, please. I think you all know that bacteriophages are highly specific and powerful and were identified as the best option by Jim O'Neill's review on antimicrobial resistance for the UK government. Next slide, please. When you look at the Thanks. When you look at the comparators, the available products that would be suitable alternatives to tackle infections, there are really only three that we can regard reasonably as active: probiotics and immune stimulation. Emerging opportunities. I think bacteriophages, of course, have been around for a long time, and of all the options available, including the active options, this is in many ways the most complex, but the most promising. Next slide, please. And when we compare them to what we've had, it could be reasonably argued that this is a third major initiative. We've had vaccines for a long time, since the days of Edward Jenner, but they give us protection from a handful of diseases, albeit with high specificity and rare toxicity. Antibiotics, of course, we've relied on for a long time, and in fact, we just about have an antibiotic for any bacteria, but we've had a lot of trouble with antimicrobial resistance, which has really defeated them. Toxicity is common, and specificity is really quite problematic, and so this is part of the damage that we're doing to human and indeed global ecosystems with antibiotics. Phages, by contrast, very high specificity, but nevertheless, it's reasonable to say that there is a phage for every bacteria. And of course, because phages evolved long before we did, it's unsurprising that since we've co-evolved, toxicity is rare, if not non-existent. Next slide, please. There's a recent review that many of you may have seen in Lancet Infectious Diseases. The statements about efficacy are difficult to interpret because there are so few well-controlled trials. And although these claims look very promising and are consistent with many people's perceptions of the value of phage therapy, I think we have to accept that it remains at the moment unproven to the level that we would normally require proof for therapies in medicine. What we can say, I think, though, next slide, please, is that phages are safe. There's no doubt that they've been used for a long time. Next, please. Next, please. Thank you. Thanks. And there's no doubt that the safety data, sorry, I've hurried you forward, just back one. There's no doubt that the safety data is clear. There is this study here, which is, as I said, published recently, Lancet Infectious Diseases, and the control groups in most of these studies have as much or greater adverse events than the phage group. Next slide, please. Okay, this has animation, so we might just do 1, 2, 3 clicks. One more, that's it. Whoops, too far, that's it. So we've treated adults and children. We've used natural phages. We've used genetically modified phages. We've used phages by themselves in groups or in cocktails, and with antibiotics and without antibiotics. We've used them by direct installation into the bladder, into the chest. We've used them by nebulization, as you see here in this picture, and we've used them mostly intravenously. And we've had good successes, and we've tried to describe these carefully and publish these in the literature so that our colleagues can review these. Next slide, please. Coming back to number 3 on the National Action Plan for Antimicrobial Resistance, Infection Prevention and Control, I think we can reasonably argue that, certainly in my clinical experience, infection prevention and control is a defensive response. It's a reactive response. When we do recognize an antimicrobial resistance problem, it is often due to passive surveillance. That is to say, it's just a finding in a clinical isolate. When we do have active surveillance in some high-risk environments like intensive care units and burns units, the response to the finding of colonization by antimicrobial resistance is principally defensive. That is to say, there's nothing positive that we can do. We simply institute barrier precautions, we emphasize hand washing, we try and focus on excessive use of antimicrobials, but we can't actually remove the antimicrobial resistance. However, many of the antimicrobial resistance pathogens are actually not normal flora. They're invasive pathogens like Pseudomonas or Acinetobacter or pathogens that you don't really need to have in your microflora. They're not an important part of human health. And they, of course, become pervasive in hospitals, and they can spread relatively clonally. And so that means that a bacteriophage that can be used proactively to eradicate either a human colonization or indeed an outbreak in a burns unit or an entire hospital can be seen as bioremediation. And this is, I think, an important argument that the number 3 of your 5 principal pillars of your national antimicrobial resistance plan can be addressed with bacteriophages. Next slide, please. The 4th principle in the national plan is promoting access to antibiotics. So given that we have such problems with resistance, and given that the principal infections which are outlined in your national plan, and indeed I think are seen the world over, Pseudomonas, E. coli, Klebsiella staphylococcus, we and many other places it can easily raise phages against these, and they can be used effectively to treat burns and wounds in hospitals and in individuals. So the 4th part of the National Action Plan is well addressed, I think we can argue, by increasing availability of bacteriophages. Next slide, please. The 5th item, of course, is new antimicrobials. Obviously, as antimicrobials become less available due to resistance, we need to have new options available. So bacteriophages, I think, we pointed out as the top of the list of alternatives to antibiotics. And importantly, we don't have time to go through the data today, and I guess it's not really the point of this discussion, but we know that antimicrobials and bacteriophages are commonly synergistic, even when they're resistant. We know, for example, that we can treat ciprofloxacin-resistant Shigella with bacteriophages inside a human macrophage and decrease the apparent MIC to ciprofloxacin, and thereby effectively restoring a drug which is otherwise lost for a pathogen which can, in Australia and increasingly around the world, and I'm sure you'll be seeing this here, can only be treated with intravenous antibiotics, including drugs like meropenem, because cefotaxime and ceftriaxone are no longer effective against the spreading clade. So this is a really important protection for the health system now and into the future. Next slide, please. I want to tell a story. If you could just, that's it, perfect. So I want to tell a little story to illustrate. We've published this, so I'll give you the reference. You can have a look at it in detail. But this is a young lady, an Australian girl. She was 9 years old. She went home to see her family in India. She was hit by a car and had to have orthopedic reconstruction in both legs. And the pins and plates in her left leg became badly infected with a very resistant Pseudomonas. It had an NDM gene, and it had a ribosomal methylase. It was resistant to all beta-lactam antibiotics, quinolones, colistin, and all the aminoglycosides. So there was really nothing to offer this young lady. She was receiving high-dose colistin and synergistic imipenem, but was really quite ineffective. She'd been on antibiotics back home in Australia for the best part of a year, in and out of hospital, with increasing pain and symptoms in her left leg. You can see the MRI scan on the top left of the screen here, showing the site of infection along the insertion point of a rod coming up through the calcaneus and into the bottom of the tibia there. So she had a very severe infection, and it was to the point that the physicians and surgeons were saying, look, all we've got to offer this young lady now in terms of control of this as amputation. And so she was notified to us, and we reached out to colleagues overseas to obtain some bacteriophage, obtained some from around near Paz in Israel, had it purified by colleagues in New York, and we treated her here. And the scan that you can see on the bottom left is taken 6 weeks later, and the photo you can see in the middle of the screen was taken about a year later. So this is a girl who was chair fast and was looking at an amputation, but very effectively treated. Next slide, please. The reason I'm telling you this story is because I want to go through some of the data with you. This slide here is a bit messy, and I apologize for that. It's quite a complicated slide with a lot of data on it. So what you can see is some lines here. The red lines are the bacterial genome count in the bloodstream. So our protocol at the moment, and certainly for this girl, was to measure the amount of bacterial DNA in the blood by quantitative PCR so that we could get an understanding of what the prey organism was doing in response to phages. The dark line, the big peak you can see, is the bacteriophage count, so that's PASA 16 genome copies per mil. And you can see that that comes a bit later as an amplification response. And you can see the C-reactive protein in milligrams per mil listed as the dotted dark lines. Yes, thank you. And so on all those arrows that you can see pointing down at the bottom of the screen, they show the phage dosing. We started off once daily, and you can see that on day 3 we gave 2 doses, and from day 7 on we gave 2 doses. And you can see the little red bars above them, the temperature lines. This young girl had a bit of a temperature soon after she had her first dose. And what I want to point out to you here is that it was immediately after the first dose that she got a spike in her C-reactive protein. She had a slight temperature of 38.5 degrees, not having been febrile before. And she got increased localized pain at the site. So we were monitoring the amount of phages in the blood, as you can see. We were monitoring the amount of viable phages, which is the dark green bars at the very bottom of the screen. And you can see 160, 105, and then 0. So we were able to detect viable phages after dosing immediately before the next dose. So on day 2, the pre-dose level showed 160 phages per mil of blood. And by that rationale, we said she doesn't need more frequent dosing. But when we were unable to detect them on day 3, we increased the dosing, and that was accompanied by another little spike in her C-reactive protein a few days later. You can see that's rather smaller. And you can see also the PASA 16 genome copies per mil. Now, PASA 16 is the bacteriophage that we obtained from Ran Paz and Ronan Hazan at the Hebrew University of Jerusalem in Israel. So what this shows, I think, in summary, is we can see a little girl who had a dose of bacteriophages that we could detect as viable phages in the bloodstream for only up to about day 5. But by about day 7, we couldn't detect viable phages anymore. I'm not going to show you these data, but in other studies we've done, we think that that's about the time we're starting to get neutralizing antibodies appear. We saw that soon after she started, she had a peak in her CRP, a little bit of localized pain, and you can see the Pseudomonas aeruginosa copies per mil. This is the sort of orange line. You see that spike up in the air, up to about 2,500 copies per mil of Pseudomonas genomes that fell again and didn't rise again until after we increased the dosing to twice a day, when we couldn't detect viable phages anymore. And you can see that this is associated with an amplification response by the PASA 16 that makes the that big spike up to about 18,000 to 20,000 genome copies per mil of phage. So we've got an amplification response that goes for some time after the phages stop at day 14. Next slide, please. So the top panel here shows a hierarchical clustering model. So what we've got here, it's a very complex figure, and again, I apologize for that, but I think it's important to show you this. And what we have here is upregulation and downregulation shown in the form of a heat map, which is the main block in the middle of the picture here. We have the genes recognized in the cluster analysis on the left as blue and red, and they appear in the panel below, which I'll come to in a second. And across the top, we have the days that the patient was treated. Now, what you can see is that the pre-phage dosing here, the response in the genes expressed by human phagocytes in the bloodstream and detected by transcriptomics were consistent with an early rise in innate immune genes. That would be the sort of thing that you might expect if you had a spike of Pseudomonas genomes in your bloodstream. And many of those genes, although they're too small to see, are typical TLR4 pathway genes. So that's the classic sort of response you would expect if you had an LPS flush into the bloodstream. So what we've got here is we have a spike of Pseudomonas DNA detected in the blood in a girl who was never bacteremic and whose bacterial DNA disappeared afterwards and never came back and had not been detected before. You have localized pain and a small rise in temperature, and this was all followed by an amplification response of the phages, evidently as they found the bacterial prey in the bone. So you can see that these all give the same message, don't they? That basically phage appeared to be attacking the bacteria, driving what appears to be a Jarisch-Herxheimer type of response, the sort of thing that you might see, you know, if you gave someone penicillin therapy for secondary syphilis sometimes, or, you know, indeed, if you treat tuberculosis, you know, we often have to use steroids to suppress inflammatory responses around the pericardium or in the brain. So it's the same kind of phenomenon, I think. And then we saw later on, at around the 10-day mark, we could clearly detect genes that were upregulated in a classic adaptive immune response. And although I haven't got the data here, this was associated with some antibodies starting to appear. So this is all the sort of antibody response that you would expect if you infused someone with a virus they hadn't seen much of before. Next slide, please. So this is only an anecdote, but it's a useful anecdote because it gave us some confidence in understanding what was going on. It gave us some confidence about the doses we were using, and it followed on previously published data in Staphylococcus aureus that was in critically ill people, and of course was much more complex to interpret. So this was a very clean analysis of a single phage in a little girl who was previously well, and so her immune response was very easy to monitor. So we in Australia, and this is a survey of Australian specialists, are relatively ready to engage with phage therapy. We did a survey, and most Australian infectious disease specialists think that if phages are prepared properly, they're safe. Many of them have patients they would have liked to have treated, but most wanted to use a trial. So everybody wants a bit of structure. Everyone's a little uncomfortable about dosing and protocols, and particularly, I think Australian doctors do not want to have ad hoc approaches to this, and this is really important. The issues that were identified were the need to have timely access, because there are big delays in getting hold of material, and to focus on a rational protocol that measured efficacy, that tested safety, that had carefully managed logistics and was properly administered. The target organisms were the ones that I think probably many of us would have expected. You know, gram-negative bacteria that were resistant bone and joint infections, prosthetic infections, you know, hip replacements, that sort of thing. And of course, the multi-resistant organisms, the gram-negatives, but in particular in things like cystic fibrosis, which is a bit of a problem in Australia. Next slide, please. Our group, we thought it was really important to try and literally get everybody engaged. And I think if you're setting up a national approach to something that is really just a new medicine, I mean, you could argue, of course, this is not new, it's been going on for 100 years, but really to us, we see this as new medicine because we don't have any of the data that we would normally expect. You know, we don't have good quality RCTs. We don't have real clarity about what dose we should be using, how long we should be using it for, all that sort of thing. So we see this as new medicine, and that means that we need everybody engaged. So we have got most of the major universities in Australia engaged. We have most of the major adult teaching hospitals around the country and every paediatric teaching hospital in the country. We have rural hospitals, city hospitals, and we have doctors and we have scientists, because this is not just about the medicine. A lot of this is about the biology. So it's really important, we thought, to have good engagement with our leading scientists around the country, most of whom are based in the big universities. So, of course, we need to engage with the public health laboratories. I'll come back to that because that's really important. And we are also sponsored by our largest state government. And we reached out to some of our colleagues overseas who've got more experience than we have in phage therapy and who have supported us in this journey. Next step, please. I'm not proposing this as any particular structure to recommend. It's just the one that we think works for us. And I think the most important point to make here is probably the heading. And that is that, you know, we don't have any funding for this. We don't have a company. We don't have any money. We're just a bunch of doctors and scientists with some research funding that we've got. And some expertise, and we all just think this is important to do, but it doesn't have a lot of funding. So it's really a coalition of people who think this is a good idea. And that meant that we had to be completely inclusive in the way that I've described it. And what we set up was a national executive group where all the senior doctors and scientists who had something to contribute. They might have been phage biologists, or they might be senior doctors who didn't maybe know much about phages. Maybe they're just experts in antimicrobial resistance, or maybe they're senior people in government health departments. But all the people that you would need, that you can think would logically be required to make this work. We had to bring this group together into a collaborative executive so that everybody had a voice, and that it was perceived as a democratic structure. So it was really important to us that this was a participative structure. We still needed to have an operational leadership, and at the moment that is really myself as director and some colleagues at my own centre, because until very, very recently, we were the only place in Australia offering phage therapy. So it was natural to start with us. But we have got now partners all over Australia and increasingly around the world. So we're really looking to try and learn from our colleagues around the world, even if they're not phage expert. They may be experts in public health or microbiology or all the other aspects that are important. Next slide, please. So at the moment, inside our country, as I said, we have no funds to manufacture. We don't even have a regulatory framework. Phages are not regulated in Australia. They're not a registered product. You can't just order them from pharmacy. And the only way we're allowed to use phages in Australia is if we can justify that the patient is likely or most likely to die or suffer extreme morbidity in the foreseeable future, and that normally means within the next 3 months, without some experimental therapy. And that means you have to show that everything has been optimized. So in Australia, we don't believe you can use phages if you've got an infection that needs an operation, but they haven't had the operation. You can't use phages instead. You have to have done the surgery that needs to be done. You have to be using the antibiotics that need to be used. And it means that we had to build a system whereby every patient who was put forward for phage therapy had to be rigorously reviewed by expert physicians. And so in Australia, in our system, you are put in touch with an infectious diseases specialist who will basically sign off that all the right things have been done. That is, the surgery that needs to happen has happened, the antibiotics that should be used are being used, and everything's being done appropriately, but the outcome is still likely to be terrible, and so an experimental therapy is therefore justifiable. So that's the rationale behind it. And as I said, we've used natural phages and genetically modified phages. Our regulator for genetic engineering is separate to the regulator for experimental therapies, strangely enough. In the US, for example, at the FDA, it is one body. I think it is the same in Europe. I don't know what it is in Nigeria, but that's an important question, I think. And we responded, of course, to our colleagues' demands for structure by developing a structured protocol. Now, we don't necessarily know the best way to treat with phages, but we had to develop a protocol by consensus, by national consensus, so that all of the expert doctors and scientists who formed our group, which, as I said, represented all the major universities and medical schools, or most of the major universities and medical schools, all of the pediatric teaching hospitals and most of the adult teaching hospitals in the major cities, we all agreed on a national protocol, which is soon, I hope, will be published in BMJ Open, and I will obviously make that available if you want to look at it. But at the moment now, we have 24 hospitals right around the country who have signed on to this as a clinical trial platform, who have local ethics governance in place. And there, as I said, in both metropolitan and in regional centres. Next slide, please. Our purpose was to standardise clinical protocols and, of course, to standardise production. Now, at the moment, we'll deal with these one at a time. The standardisation of clinical protocols meant that we had to propose an ideal dose. So again, we don't have time to discuss the rationale for all this, and we have published some of this, and it will, as I said, it is stipulated in our protocol, which we can make available if you wish. But essentially, on the basis of published animal models, we rationalised that for someone with severe bacteremic sepsis, and in studies that we've done, we know that the typical bacteremia is between 10 to the 3 and 10 to the 5 CFU per mil in septic shock, then you would need to administer about 10 to the 9 viral particles diluted into, say, a 5-litre blood volume to have a reasonable likelihood of a multiplicity of infection of around 10, which animal models suggest is what's required. So that's the rationale for that dose. The strategy that we used, as I said, we've used in staph and we've used in the Pseudomonas case that I mentioned. We've done it in about 20 people now and studied all of the pharmacokinetics and dynamics, and we believe that that is actually quite a rational dose. I don't know how long to treat for. It's certainly true that we've treated some mycobacterial infections for months, but mostly we believe a 14-day course is rational, maybe shorter. I mean, we modelled this on what we think a rational antibiotic course is. As I said, we triage this through specialist physicians. we have this trial framework, and that's important because if we run this as a clinical trial, it means that every hospital that signs on is subject to the national sanctions that apply if you deviate from a clinical trial protocol. So if we just gave people a protocol and said, here, do it this way, we are likely to experience the same thing that our colleagues in Brussels experienced. So Jean-Paul Pirnet told me not long ago that he has given out probably 100 different phages now, and he estimates that he's had data back from about 15%. And those data are not structured, so you can't compare them. Even the tiny amount that has come back, it's very difficult to compare and to learn from. So we've stipulated this structure, and the sanctions that come with the clinical trial network in Australia means that if people don't provide the data, then they're sanctioned by their hospital. So we think that's important. Production standardization is also important. Most people will produce a phage genomic safety data description of some sort. And normally what you're looking to do is to guarantee that the phages don't have capacity to integrate into bacterial genomes, like the cholera phage does, or the prophages that cause diphtheria. So obviously you don't want to use that kind of a phage. You have to use an obligolytic phage, and you can do that from the genome. So that's something we believe has to be provided. And every manufacturer who sends you a phage, you would expect to have that information, I think. We also believe that the bacterial production strain, you need to do the same thing. And the reason for that is that although you can, in your bacterial safety data sheet, demonstrate that your product is clean, that it's pure, that it doesn't have much endotoxin in it, you could theoretically co-purify a protein toxin from the bacterial production strain and administer that with the phage unless you specifically measure it. Many people who are producing phages at the moment use a monocyte activation test along with their other tests for batch safety to give some assurance that there's nothing else in the way of pyrogens. But I don't think we could be absolutely confident that every possible bacterial toxin that could be co-purified, I mean, if you wanted to treat cholera with a cholera phage or diphtheria with a diphtheria phage, you would probably have to grow it in the strain that it naturally targets. And because you're targeting pathogenic strains, you may be growing it in a pathogen. And of course, you may therefore co-purify a toxin. So we think that's important, and the best way to do that is just by doing a bacterial genome. So we do that, and I think that's a little different to what others do, but that's what we do. We also produce, of course, a batch safety data sheet, so a pH, a purity and sterility. Obviously, you want to make sure that it's a clean, sterile product. We do an endotoxin assay, of course, because we want to make sure that we are producing less than 5 endotoxin units per dose. If we want to administer that dose, that's per kilo per dose. And that means that for a 70-kilo man, less than 350 endotoxin units, which is relatively straightforward to achieve. And we do endpoint genomics because we don't want co-purified prophages, which can be a problem in some production strains. So you might not have bacterial toxins, but you might get a co-purified prophage. And of course, we produce a matching diagnostic sheet, which contains a spot test and a kinetics test. Next slide, please. So we do, as I said, our standardized therapy and monitoring protocol. We think that's really important to build trust and to ensure that everyone's doing the same thing and we can measure what we're doing. Next slide, please. We've chosen a number of clinical trials that we will now seek funding for. We don't have funding for them yet, but we will seek funding for them. And they are the ones that I mentioned before that we have identified as most important. And we're trying to build them in as adaptive platform studies on existing adaptive platform studies, which is kind of the easiest way to do a modern trial, I think. Next slide, please. I think I've probably got too many slides and I might ask if we need to stop soon, but let me just go for a couple more slides before we pause. I think this is an important point. One of the problems with production of phages, of course, is that they're so specific that you risk purifying a phage to treat someone that you will never use again. So you need to know which are the likely phages, and you can do this if you target your national surveillance program. So if you know the bacteria that commonly cause infection in your hospital or your city or your country, you can identify the phages that you are most likely to need. Next slide, please. Yeah, and then we do just endpoint quality control in the way I described a second ago. Next slide. And this is important because there is a point at which you become efficient. And if you can use at least 10% of a batch, and a decent batch might have a shelf life of 6 months or even more, then you can imagine how you might do that. You can imagine how you can easily get economies of scale. And if you do that, just click on a bit, please. Then we think you only need to treat about 6 to 8 people every 6 to 12 months in a batch to get down to doses that are equivalent to our current rescue antibiotics. We think we can get phage production down below $5,000 a course. Next slide, please. And that is, of course, for purified intravenous phages. Obviously, if you want to use them topically on a wound or something, it's much, 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 much cheaper. So we argue this is an efficient spend of public health dollars. Can you just click on a little bit, please? Thank you. One more. So we compared the bang for the buck, if you like. So we know some things are expensive. Intensive care is really expensive. Bone marrow transplantation is really expensive. But the amount of people you save with them in a country like ours means that you get a lot of value for money. So the quality adjusted life year costs are very low. And phage therapy is like that. It is a cheap way to get good health returns. Next slide, please. So I think this is probably the point at which we should pause because it's now 1:45. I just wanted to look at this question about whether, whether this is comparable. I mean, your country is much bigger than ours. You have a much greater population than ours. You have a younger population than ours. And although your population is urbanizing, it's still more rural than urban, but you have bigger cities than ours. We have a national surveillance program, and we think that we are seeing the same kind of bugs that you see, or at least you've identified in your national action plan, but we know you have more antibiotic resistance. Next slide, please. So just one more, next slide. Yeah, and yeah, I can finish up now, I think. So if you want to find out more about us, you can just go to Google and write in Phage Australia or Phage and Australia or whatever, and you'll soon hit us. Next slide. Next slide. That's it. Yeah, so we are at the moment looking to partner with others. We've got a strong partnership now with Canada. We've got strong partners in the US and Israel, and we'd love to have more partners. We are able to sequence strains if people want to submit them, and phages if people want them sequenced, and list them in a biobank. And we want to make sure that people's ownership of those phages is protected. So wherever they come from, that becomes registered in the biobank. You can see all this by going to our website. Next slide, please. And yes, we're just closing off now. So I'd point out that we've already started to have lab meetings where we're talking a bit about the biology of phages, and a lot of the scientists are talking through their issues with purification or biobanking or anything. And we're just about to start some clinical rounds with some of the colleagues that I mentioned, ran their PAS at the Hebrew University in Jerusalem, Greg German at Canada, Public Health Canada, and Gina Sa at the Mayo Clinic in the US. So look, I think I should stop here because we're going to run out of time for questions, and thank you so much for your attention.
Thank you very much, Professor Jon Iredell. It was a very good talk. I especially enjoyed the level of detail and animations that your slides had, and the very beautiful graphics. So thank you very much. I also like to say that we can drop our questions in the chat box, or just identify by raising up our hands. So one of our faculty members is here in person of Dr. Veronica Guneye. I just want to use this medium to recognize our presence. So Nutin, you can speak up now. Nutin, are you still here?
Might be on mute.
Sorry. Sorry, there's some noise here first. I need to reduce it.
Okay, no problem. So others can drop their questions in the Q&A or chat box.
Yes. Thanks, Professor Iredell, for your great presentation. I know you have a great team there and expertise to set up a Phage biobank on your side over there. So I think some African Phage researchers are also trying to do the same thing, and I'm trying to see a little bit the rationale behind the fact that we should come up with data on the bacterial production strain. I mean, you talk about genomics data to prove a little bit the efficacy, not the safety, instead of the Phage lysate, because you have been talking about normally when we are producing the phages in the host pathogen or the host bacteria. It can be actually the phage can be, maybe we can get the phage with the bacterial toxins. And when you are talking about coming up with genomics data using the bacterial production strain, are you actually referring to the toxin genes or the whole bacterial strain genome? That's what I would like to know.
Yeah, that's a really good question. It goes to the way we describe them, actually. So we haven't talked about that side much, but some of our colleagues, particularly Nuri Benzakour, who's a senior bioinformatician who works with us at the moment, has been working to develop an annotation system that better identifies the things that we think are relevant in that regard. So, for example, in our public health laboratory, if we do genome sequencing, we will try and produce something that tells us whether things are related. So if there's a TB outbreak in 2 different states, or a salmonella outbreak, we might do their genomes and talk about relatedness so that we can understand where it came from and how to stop it. But when we're talking about phages, our concern is that if we want to create a phage against, say, a strain that causes hemolytic uremic syndrome, we really need to understand whether we might be co-purifying the toxin that causes the problem in the first place. And I guess that goes to the question of, well, what do you do with that information? I think we need to first assure the regulator and the prescriber and the consumer that we're not administering co-purified dangerous toxins. But secondly, of course, I guess it means that logically we would seek strains that are less toxic that might also host the phage and allow us to use them as production strains. I think it's an obligation that we probably should accept. You may be right. Perhaps perhaps it'll turn out to be a bit unnecessary, but I guess time will tell.
Thank you very much, Professor Iredell. So someone also has a question here. Okay, so she asks, Amrita Salim, sorry about that, asks that I have a general question. What are the ideal features of a production host? So a phage production host, that is.
Yeah, good question. So I guess from my point of view, I mean, I don't have any unique perspectives, but I think most people obviously want to see high levels of productivity. Ideally, a host that is not giving you a whole bunch of co-purified prophages. Ideally, a host that is not going to give you co-purified toxins. And you know, it might be argued that given that we spend a lot of time and effort getting rid of LPS from our phages, that maybe we should be looking at ways to reduce the amount of LPS that is naturally present in the production host. I think at the moment, though, often because of the need for LPS receptors for natural infection, that's a bit tricky. So I think in summary, it's really just a host that doesn't produce toxins or cophages, but does generate a good yield of phages.
Okay, thank you very much. So we'll take one more question. Dr. Shafiq, you can speak up.
Thanks, Jon, for a good presentation. I just have a comment regarding the genomics of bacteriophages, because in our experience, there are a lot of proteins in the bacteriophages for which we not know that either what type of proteins they are. They're just showing like hypothetical proteins. But when you are applying the bacteriophages, do you do some analysis that either those proteins have some toxic effect on the cells or not, or what we can do about those proteins whenever we are selecting some phages for therapy?
Yeah, that's a really good question, Shafiq, and I don't have a really good answer, I'm afraid. Like everyone, like you, I think we're still trying to understand a lot of the genomic packages of these bacteriophages, and I guess that's one of the reasons why we think it's so important to have phage biologists really centrally involved in what would otherwise seem to be a relatively clinical story. I don't think we can do this just as a medical story. Really, there's so many biological questions, as you point out, that we really need to do this as a medicine science collaboration. So I'm really lucky where I am. I've got some fantastic scientists who work with me. And we've also got some fantastic scientists as collaborators who've been really generous in providing their expertise, their knowledge. And I think, I mean, you're in the same position as us, really. I think you've got the science, you've got the medicine, and we need to marry them together. But I think, yes, it is true that some of the phages will contain proteins that will be problematic, and we don't know quite what they are yet. We know that phages do have an interaction with mammalian cells. We think the evidence is that that interaction is almost invariably safe and non-problematic. But yes, there are some important unknowns there, and I don't have a good answer.
Okay, thanks a lot.
Okay, so thank you very much for your good presentation, Professor Jon Iredell, and to all the participants present and those that asked some questions. I really enjoyed the presentation so much, and it's one of my favorites so far. Maybe I'm biased because I'm the one moderating, but it has been very good. So thank you very much. We'll take a 3-minute recess before we start. Okay.
Thank you very much for having me. Bye-bye.
Yes, thank you very much, sir. Okay, so I'll be passing the mic to my co-moderator for today, in person of Daramola Oluwasegun Isaac is the Phage team leader for Ibadan Bacteriophage research team. He will be taking the next session. We'll be moderating the next session.
So before, good morning and good day, everyone. So before we move into the last session for today, which is that by Dr. Jinali Ngala and Ivy Mutai, we just want to use about 2-3 minutes to sensitize the attendees here about our plan to launch the second edition of our team's bacteriophage journal. So we had an issue out last year, and we plan to also have a renewed edition of the familial journal. So what's been displayed here is the cover of the previous edition, which is still currently available for download in the PDF format also. So this year, I would like to inform us all that we have plans towards launching the second edition, the second issue of this journal. And once that is done, by November, we would have that communicated to the population of Phage community at large, and then we can have access to abstracts being published from the team and outside of the team. So we would have or we would all have access to it to be on our page on our website and on Twitter also, and all our social media platforms. So we are currently working on having that ready after the World Phage Week, also in collaboration with the celebration and commemoration of the International World Phage Day. So we hope to have that soon. Then we would now